Welcome back to the Health Longevity Secret Show with Dr. Robert Lufkin. Glycans are being hailed as possibly the next great biological longevity clock. Today, we speak with the leading expert on the subject. Dr. Gordan Lauc is a professor of biochemistry and molecular biology at the University of Zagreb and co-founder and chief scientific officer at GlycanAge. He graduated from the University of Zagreb Faculty of Science in 1992 and obtained a PhD in biochemistry at the University of Zagreb in 1995. Health Longevity Secret Show listeners who wish to try the GlycanAge longevity clock on themselves can receive a 20% discount by entering the code Robert Lufkin, no space in the middle, at the GlycanAge website, and we'll have a link in the show notes as well. Now, please enjoy this interview with Dr. Gordan Lauc. Hey, Gordan, welcome to the show.
Hi, thank you for the invitation.
I'm so excited to learn with our audience today all about glycans from really the world expert on the subject. But before we dive into that, maybe we could just take a moment and tell us a little bit about how you came to be so interested in this fascinating area.
Well, it was actually more an accident, which happened 29 years ago. I was doing my PhD, and at that time, my topic was physical biochemistry of an enzyme, and I was purifying a protein. And experiment failed on Friday afternoon. And at that point, I asked my boss, okay, I have nothing to do over the weekend, give me something to do. And he came with a brand new set to analyze glycans, which was never done before. And I did it first time nearly 30 years ago, and I keep doing it for the last 30 years. It's a lot of fun.
That's great. I've heard so many interesting things about the subject. I'm really looking forward to this deep dive. I've heard glycans as described as maybe a good kind of sugar to have in an era of low carbs. I've also heard your great paper in Frontiers in Genetics that you wrote a few years back on glycans, the third revolution in evolution. So perhaps we could start off by just telling our listeners about what the first two revolutions were, and then how glycans are the third revolution.
So life on this planet began as a RNA world. So ribonucleic acids were the first molecules which were containing information and replicating this information. And then as evolution was happening, ribonucleic acids recruited amino acids to make proteins, because for nucleic acids, there are only 4 building blocks. They're relatively similar, very hard to make many different structures. For proteins, we have 20 amino acids, so there's a lot more information which can be stored there, and the number of structures which can be made is very different. But then, when the life became multicellular, we became more and more complex. And when we look only in genes, and every protein is encoded by a gene. So for every protein, we have a gene which we inherit from our parents. Bacteria, which is the simplest organism, would have around 5,000 genes. And even when I was studying, we were taught that humans have at least 100,000 genes, because we are so much more complicated than bacteria. But when we sequence our genome, we learned that we have approximately 20,000 genes. So humans are only 4 times more complicated than bacteria, and we have trillions of cells. These cells have to differentiate, they have to talk together. And actually what enabled creation of multicellular life was invention of protein glycosylation. So glycans are made in a process called glycosylation. They are not based on a genetic template. So you don't make a glycan based on a template in your genes, but it's in a complex network interaction of hundreds of genes. And these structures make our total set of different molecules several orders of magnitude larger. So we have approximately 20,000 genes, maybe around 100,000 proteins, but we have tens or hundreds of millions of different glycoproteins. So this is why we called it the third revolution in evolution. The first was RNA, the second were proteins, and the third are the glycans.
Wow, so let's see if I can summarize that. So the DNA, as we learned about, has 4 base pairs, and it's a linear arrangement of information. Each DNA, each gene sequence specifies 1 protein in some cases, not all of them, but yeah, more or less. And the proteins are encoded by 20 different amino acids, also in a linear fashion. And now the next revolution is a layer of information on top of the proteins that is the glycosylation. And I understand what you're saying, that is not linear, that nope. In other words, it's exponentially more complex. And that's yes. the beauty and the power of glycosylation as containing information.
So we estimated there are approximately 2,000 different glycan blocks, which can be added to proteins. So you can have 1 protein and then attach tens, hundreds of different glycans, and there can be multiple glycans. So even for simple glycoproteins, a single polypeptide sequence, this is the protein part, can be converted into hundreds of different glycoproteins by adding different glycans. So there's a huge layer of information which we are all using in different ways. We use it for our immune system, we use it for development, we use it in our brain function, and so on.
So is it fair to say that on the show we've had several speakers talk about epigenetics, and we've learned about DNA methylation and histones and RNA as ways of modifying epigenetic modification of DNA. What you're saying is this is almost a sort of epiproteinomics, if you will. It's a glycosylation of the proteins in a much more complex fashion, potentially, than the epigenetics. But is it fair to say that this epiproteinomics is also influenced by our environment in a similar way that the epigenetics is? So it's a reflection of our lifestyle and what happens to us in our lives.
Yes, but actually even more so, because for glycans, there are 3 key components which decide about their structures. One is genetics, and most of our glycome composition is between 30 and 50% genetically encoded. So people are different and their glycans are slightly different. Then there is a strong epigenetic component. So epigenetics is kind of memory of past events. What happened before in your life gets encoded in these epigenetic modifications which make your genes more or less active. And then there is a third component, and this is direct environment. Epigenetics is also a bit affected by direct environment. If you eat special food, you get more or less methylation and so on. But for glycans, it's even more emphasized, because glycans are chemically related with sugars which we eat. And this energy metabolism is directly talking to the glycosylation. So people who eat more carbohydrates will have a little bit different glycans than people who don't eat carbohydrates. So also the current environment, the food, actually, when the thinking we do is affecting glycosylation. So 3 components: genes, this is our memory from millions of years; epigenetics, this is our lifetime memory; and direct environment affect the glycosylation and decide about the individual glycans.
And back to the comparison with epigenetics, again, one of the for many people, non-intuitive aspects of DNA methylation as a mechanism for epigenetic modification is that with time with aging, it's not merely the increasing accumulation of methyl groups to modify the DNA, but it's rather a rearrangement of the methylation groups, and there's no net methylation differences in many cases, but it's specifically the way they're organized. Is that true with glycosylation with aging? In other words, is it's more a rearrangement, or is it just as you get older and older, you get more and more glycosylation, like rust on a, on a iron bridge or something?
So both epigenetics and glycosylation is information. So it's not only the amount, it's the arrangement, it's the specific sites which either methylated or not, and the same is for glycosylation. So glycosylation is a creation of specific structures. Each of these structures contain information and perform function. Something what people often confuse is glycation and glycosylation. So glycation, like HbA1c, or somewhere they call it only I think 1c or whatever. Usually Americans have these short names. So HbA1c is a glycated hemoglobin. Hemoglobin which reacts with too much glucose in your blood, creates this HbA1c. And this is a process which is chemical, which has nothing to do with information. It's just a biomarker of too much glucose in our blood. Glycosylation has nothing to do with that, besides being chemically related. Glycosylation is a process which is regulated on multiple levels. For example, for immunoglobulin G, which we study a lot, these are the key antibodies we have in our body, we know that it's over 40 different genes which work together to make a decision about its glycosylation. And these genes are very important for different diseases, and people who have kind of glycosylation changed in a way to be more pro-inflammatory, they will develop these diseases easier than people who have glycans which are more suppressing inflammation. So these are functional parts of a protein which do a specific work.
Before we get into the inflammation, let me underscore the point you just made that when we're talking about glycans and the sort of epiproteomics, if you will, that's glycosylation, and that is different from glycation, or the AGE that our audience has heard about for advanced glycation end products. That's different than what we're talking about here, correct? That's glycation versus glycosylation.
Yes, these are two completely different things. The glycosylation is a regulated enzymatic process. Glycation is a chemical reaction which tells you that there is too much glucose in your body and that somebody is on a path to diabetes.
And glycation is almost like rust on the, it's exactly, whereas
it's exactly like rust, yeah.
Glycosylation, on the other hand, is information storage, and it's directed it's directed information. Now, onto inflammation. Is that the primary longevity risk factor that glycans encode, or are they associated with other types of aging phenomena or chronic disease phenomena, or is it all manifested through primarily inflammation markers?
So if we talk about glycans in general, there are so many different glycans and so many different proteins that this is not possible to generalize. But what we do a lot of research on is glycans or immunoglobulin G. Immunoglobulin G is the main class of antibodies, and we know that its activity is regulated by glycosylation. So there are specific forms of glycans which can be added to IgG, which then direct it to one type of receptors, or different glycans directed to other type of receptors. So this single molecule can be either pro-inflammatory or anti-inflammatory, depending on which glycans it has. So immunoglobulin glycans are one of the key regulators of inflammation. And we know for a long time, for example, If you take immunoglobulins from a young and healthy person, it can be used as a drug to suppress inflammation in some inflammatory diseases, or even a long time ago in former Yugoslavia, communist politicians were using it just to extend their lifespan. They were giving young immunoglobulins to make them feel better because they are suppressing inflammation. And by suppressing inflammation, they save so much energy, which we normally spend for low-grade chronic inflammation. Because if we go step back, what is inflammation? So inflammation is a process of rebuilding something which is wrong. Normally, we activate inflammation if there's some kind of infection. So there are bacteria growing in part of our body. Our neutrophils come in, they kill everybody, everything. They just shoot poison on that area, kill the invaders, and then you rebuild this part of your tissue. So we all know, when we get a small cut or some kind of infection, then we have a red swelling. This is blood coming in, killing everybody. And then this disappears in a few days. So inflammation is good if it is acute. But what happens with aging is that there are so many small inflammatory processes everywhere in our body that it is first consuming huge amount of energy. And second, it's making many small scars. So each inflammatory process, there is rebuilding, and not everything is being done perfectly, and too much inflammation generally leads to different type of damages. And it is now generally believed that inflammation is underlying most chronic diseases, which are the main burden today: cardiovascular, cardiometabolic, inflammatory diseases. Even people now think that Alzheimer is inflammation of the brain. So definitely, since we are living for as long as we live now, and again, we have to remember that biologically, there is no selective pressure to keep us alive after the reproductive age. So all the biological mechanisms were optimized to keep us alive while we are producing children. When we stop producing children, we are just living kind of a bonus time, which we got by healthy living, sanitation, medication, and so on. So there is no evolutionary pressure to keep neither of us alive at the moment, because we've done our job in making babies. And then inflammation kind of gets loose, too much inflammation. And this is also result of too much food. We mostly eat too much, way too much. We could survive with much less food. And also the sedentary lifestyle, this all promotes inflammation. And this too much inflammation is believed to be one of the major drivers of aging. There is a process called inflammaging, which is kind of a vicious circle of inflammation, more damage, more inflammation, which then ends up in a disease. And glycans are very important there. So if we have immunoglobulin glycans which are suppressing this inflammation, then we slow down aging, we have less risk to develop different chronic diseases. Generally, we feel better and we are younger.
Yeah, it was certainly an epiphany for me, learning how inflammation is, like, as you say, it's underlying essentially every major chronic disease we face, and that things like brushing our teeth to decrease dental inflammation can actually lower our risk of Alzheimer's disease and dementia and heart attacks and other things. The other fascinating thing, what you mentioned about inflammation, was that it's all not good or bad. In other words, that acute inflammation is good and necessary for handling certain types of attacks on the body, but chronic inflammation is bad. And that seems to mirror what we're learning about stress also as a longevity risk factor. In other words, acute stress, hormesis, saunas, ice baths, exercise, those acute stresses are actually beneficial and healthy. But chronic low-grade stress with glucocorticoid elevations, that is the dangerous, damaging part. So it seems to be stress and inflammation have that same feature, that the acute is good and maybe even necessary for survival, but the chronic, indolent, low grade stress or inflammation can be very damaging. And a large part of the inflammation, you say, is mediated through the glycosylation of the IgG that you're seeing on that?
Yes, we definitely see, for example, if people have an inflammatory disease, they will have more of these inflammatory glycans. And in most of the diseases, we see changes in glycans 5 to 10 years before the disease develops. Because, you know, disease is a kind of an endpoint. So something is so badly damaged that we feel pain, that something is not working, and then we ask for help. We go to physician, and then we get some kind of a diagnosis, meaning a name for our disease. Diabetes, ulcerative colitis, arthritis, whatever. But, you know, this did not happen in a minute. This did not happen in a day. This was developing for decades, and our body is made to compensate for all the problems. And for a long time, it was compensating, and then at one point, it couldn't anymore. And usually, this is the point of no return. So once you have a fully blown, I don't know, Crohn's disease, there's no cure. You can just alleviate the symptoms, you can make people feel better, but they will be sick for the rest of their life. But this is why we call it a chronic disease. And the idea which we and many other people have is, let's try to find these processes early. Let's identify it at the level of only a subtle molecular change. Some molecules are not functioning properly, because then we still have plenty of time, maybe 5 to 10 years, to modify lifestyle, take some kind of a preventive therapy, and avoid disease. Do not allow it to fully develop into disease. And this is where we think glycans can help a lot. Because, for example, if we take the main killer today, these are cardiovascular diseases, we can see glycans changing 5 to 10 years before people develop hypertension, because before people get a heart attack, before they get stroke, we see glycans already changing. And we also see the same thing for diabetes, but different glycans, not the same glycans as for cardiovascular diseases. And we can use it as a kind of a motivator to help people change the lifestyle. Because we all know what are the healthy lifestyle habits. Now, if you don't eat too much, this is healthy. If you eat too much, this is not healthy. If you exercise moderately, this is healthy. If you exercise too little or too much, this is not healthy. If you sleep enough, if you're not under stress, these are all healthy things. But it's so hard. You know, it is so hard not to be under stress, not to eat, to regularly exercise, and people generally don't do it. And they don't do it because the bill is coming so late. No, I will pay for my mistakes in 20 or 30 years. I'm not going to skip that cake because of something what will happen in 20 or 30 years. But today, with all these smart things which are tracking us, or with the molecules which we can measure, you can get much feedback much sooner. For example, we have this GlycanAge test of biological age, which was developed in my lab. And of course, I do have some commercial interest in that. So you have to judge what I'm saying. I'm definitely impartial here. I'm not impartial here. We know that if you live a healthy lifestyle, these glycans will improve. And when I see on myself that when I lose weight, my GlycanAge is going down, this is helping me to motivate myself to control what I'm eating in the next few months. So it's, I think it's definitely one good way of measuring what is happening and how what I'm doing at the moment is affecting me at the molecular level. Because this is something I cannot see in the mirror.
Yeah, that's such an important concept. The idea of biomarkers or tests being motivational factors for helping us stay on the right path in our lifestyle. And the other point you made is game changing in medicine, that we're seeing that diseases that we used to wait until they, like dementia, you wait until people have cognitive impairment, but we now know that there are changes in parts of the brain like the hippocampus that can be measured with an MR scan, and you can see hippocampal atrophy or loss of volume in that 10 years before the Alzheimer's disease presents. And so people can be motivated to change that. And like you say, with diabetes, if you wait till the glucose rises, which is the diagnostic criteria for diabetes, we know that 10 years before the glucose rises, the insulin has already become abnormal, but most people don't check fasting insulin. But with a test like that, or with GlycanAge, we could provide that information to people and really motivate them. So let's talk about clocks and biological clocks, and we've talked in other presentations about the value of longevity clocks for motivating people for longevity programs. How does a glycosylation clock differ from a DNA methylation clock?
So we came to that point from completely opposite direction. So we were in my lab, and I have a large lab with over 50 people working in it. We were interested in what happens with glycans in people who have a disease. And we already analyzed over 150,000 different people. So from different cohorts, people were studying different diseases. I just emailed them, they sent me a sample, we analyzed the glycans. And then first thing we learned, glycans change a lot with age. Because we just realized we cannot take, I don't know, healthy people which have average age of 40 with sick people of average age of 60 and compare them, because they will be different just because of age. And then we modeled chronological age using our glycans. And we can guess somebody's chronological age with an average error of 9 years using glycans. And we did it first, when we did it first time, it was actually very interesting. We were thinking about the forensic applications, that we could identify how old was a person who left a drop of blood on a crime scene, because I also have a forensic lab, so this was an interesting application. But then DNA methylation came in, which was way more accurate for the chronological age. So Steve's epigenetic clock, Steve Horvath's epigenetic clock, can guess an chronological age plus minus a year or two in a population cohort. And then we realized, actually, this is not fun. If you can accurately guess the chronological age, where is the valuable information there? Because I know my age. I don't need a molecule which will tell me my chronological age. And then we learned that the difference between the GlycanAge and the chronological age is actually, by large, determined with a lifestyle. So people living healthy lifestyle will have a lower GlycanAge. People having unhealthy lifestyle will have it higher. Also disease. People with different diseases will have a higher GlycanAge than healthy people. Steve also realized the same issue, and then he developed the Grimm clock, and now there are many other epigenetic clocks which are not tuned for chronological age, because that's not fun, but trying to give some kind of biological information. And I like epigenetic clocks, they're great. The only problem with them is that each of them is measuring something differently. And also with all the companies selling the epigenetic clocks, each of them is a little bit different. They all call them epigenetic clocks, but they measure different CpG marks. So we will have to actually perform independent research for each of these clocks to know what they actually measure. So for glycans, since we started from the opposite direction, we know that high GlycanAge means more chronic inflammation, And these molecules are actually functional effectors of inflammation. So if you give immunoglobulins with these glycans which suppress inflammation, they will suppress inflammation. They can be used as a cure to heal people who have too much inflammation. So once we measure glycans in a person, and if we see bad glycans, we have no problems in telling a person, no, you have bad molecules which are doing damage to your body. Maybe you can try to change something. And then if we see more and more good glycans appearing, we can say, you know, this is definitely helping you. While for epigenetics, we don't really know what each of the sites is doing. So epigenetic is information, and as time is progressing, we'll have more and more knowledge about it, and it will be much more useful. And I'm actually pretty certain that if we look properly at epigenetics, we'll find a proxy for the GlycanAge in epigenetics. And actually, this is something what we are now working together with Steve Horvath to compare glycosylation and methylation in the same people. But at the moment, I think these glycans give more actionable information. And the second big difference is that one of the disadvantages of epigenetic clocks so far was that they poorly react to interventions. There are only few papers showing that if you do very aggressive therapy, epigenetic clocks changes a bit. To the contrary, we are seeing glycans changing a lot with relatively simple interventions like a weight loss. People losing weight are improving their glycans, people gaining weight have their glycans going in the bad direction.
What are the most, well, yeah, to emphasize your point, yeah, it seems clear that beyond the first generation forensic type chronological age methylation clocks, we're now seeing specialized ones, and it depends on what they're trained on, like Morgan Levine is showing that you can really train them on anything, and depending on what you train them on, that's what they will be most sensitive to. So what you're saying with glycan clocks, they're at least the ones for the IgG that you're using are predisposed to inflammation, but inflammation is at the basis of all our diseases. So it's very, very useful things there. So which interventions have you seen that are most effective at reversing or increasing the GlycanAge that you calculate?
So here we have something which is anecdotal evidence, meaning you have a few people, 50, 100, who do something and then we see a change, and then there are some proper studies which we have done. So anecdotally, we collaborate a lot with one anti-aging physician in New York with his PhysioAge platform, and most of his patients will become 20 to 30 years younger after approximately a year in a practice. And I think the strongest thing he's giving to them are hormones. He calls this hormonal optimization, and definitely in women around perimenopause, estrogen is a big issue. Once women start to lose estrogen, their GlycanAge goes up drastically. And for men, it's also estrogen, but in men, estrogen is generated from testosterone with an enzyme called aromatase. And we did one proper placebo-controlled randomized trial where we had a group of both men and women, where in woman menopause and also in women it's called andropause probably, gonadal hormones were blocked chemically. So you have a young woman and you block the production of hormones, and then there was a treatment group where they were receiving estrogen supplementation, and placebo where they were not receiving anything. They were just given a patch, but it was a placebo patch. And we saw that in a placebo group, women on average aged 9 years in 6 months. While an estrogen group, this did not happen. So women who were given estrogen back were protected from this accelerated aging. And we see it often in women in the perimenopause, that somebody who had, for example, a very good GlycanAge before would age in like 20, 30 years in a year. Once given hormone replacement therapy, this goes down again. In men, testosterone works unless you block aromatase. If you block aromatase, they're inhibitors of aromatase, because people when they give testosterone to men, they want to see very high testosterone. And then testosterone is usually being lost to estrogen. And this is why some people give this supplementation, this blocker of aromatase to keep testosterone high. But actually, this higher estrogen is what also protects men from these bad pro-inflammatory glycans. So definitely one thing which works is hormones.
It's interesting that because of the ITP results for the interventions testing program, which is where mice are given certain drugs, and then they actually measure the longevity in the mice, how long they live, and only a few drugs have been positive. Things like rapamycin have been effective in lifespan extension, but one of the other drugs is 17-alpha estradiol, which at least in men it doesn't produce sex characteristics, but it's again related to estrogen and all.
I know that study. So we have problems with working with mice. We have one study where we compared, I think, close to 100 different strains of mice, and they're so different because, you know, all the mice which we have in a lab are actually immunological imbeciles. They have lost most of the immune processes because the only thing they need to do is to eat and make little mice. They don't have all the challenges which normal mice in the wild has. So mice are not such a good model for this glycan aging. At least, well, some strains of mice are, but main laboratory mice are not as good. And yes, estrogen is very important. And something which is maybe simpler and maybe more acceptable to majority of people is also weight loss. We have very strong data showing that if you lose weight, your glycans improve. For example, we had several cohorts of people on bariatric surgery. So they had this stomach reduction, they lose 10, 20, 30 kilos. And we even have one client who improved for 37 years in half a year. So not everybody improves so much, but weight loss helps. But again, bariatric surgery is quite radical. So we were wondering whether just the regular weight loss is also helping. And we had a study on 2,000 twins, which were analyzed 3 times over 15 years. We had 3 samples over 15 years. And we have seen there that twins who were gaining weight over time were aging faster based on a glycan clock than twins who were losing weight. And also we have some people who went into different programs, and losing weight generally reduces GlycanAge, with one exception. If you're exercising too much, if you're overtraining to lose weight, then you're also increasing inflammation. So it's not good on the GlycanAge test. So too much exercise is not good.
Yeah, so you were seeing, because I've heard anecdotal reports about athletes doing biological clocks, and they find their age is elevated when you think they're, you know, healthy athletes, they would be younger.
But not all anecdotal. We have several studies, properly designed studies, which we did, where we see that if people overtrain, they actually get worse. And we also have one proper study where we did this high intensity interval training, where they immediately improved. So I think training is important, but relaxation is equally important. And although, you know, well, I think this was actually brainwash in the last 30 years by the food industry, where to avoid scrutiny of the processed food, they were forcing, they are kind of supporting researchers to study exercise. And then the mantra was, you just have to sweat it out. You have to exercise more, and then you can eat all this junk food which we are selling you. And now we actually know these two things don't compensate. So you cannot sweat out the junk food. You have to stop eating the junk food, and then you have to exercise moderately, and this is fine. And this is so logical because, professional athletes, they're old, in their 30s. They're weird out because they exercise all the time. We don't have bodies which can work all the time. We have to exercise, give this stress, give this signal for muscles to build up, for everything which happens when you exercise, but then we have to relax and allow our body to fix all these small damages which happen.
Now we have a glycosylation clock now that measures different aspects of our lives, but it's strongly influenced by inflammation. And we've shown that lifestyle and other interventions can reverse or dial back the age on that clock. Now, I guess the final question is, what evidence do we have, or do we know that reversing the age on a glycan clock shows any benefit? Is it beneficial? It's sort of like I may have wrinkles in my forehead, and that's a marker for aging. But if I take Botox and inject them, sorry for this analogy, I live in Los Angeles, so we see a lot of that. But if I inject here, then the wrinkles go away. So that biomarker for aging, I look younger, but really none of the fundamental processes have changed. It's sort of an epiphenomenon of that. So I guess the question, anytime we have clocks that we rewind, the next question is, do we know that rewinding improves longevity or decreased risk of disease, or is it just too soon to tell?
So we have one very nice, elegant study, which we did together with Phil Scholl from the University of Texas Southwestern, where he has a line of evidence suggesting that these bad IgG glycans, which lack this final salicylic acid, are actually causing hypertension. So he did a series of studies on mice, where if you put mice on a high-fat diet, they become obese and they develop hypertension, and their glycans become old. And there is a dietary supplement called N-acetylmonosamine, or MANAC. If you give it to mice who are on a high-fat diet, they become obese, but their immunoglobulin glycans stay young. And mice who were on a only high-fat diet developed hypertension, while mice who were supplemented with the monosamine, they did not develop hypertension. So actually, this monosamine, which is a sugar, but sugar which is causing this good type of IgG glycosylation, prevented mice for developing hypertension. And this was published in Circulation, which is the best cardiovascular journal. And we joined that study by looking at thousands of people. We didn't do intervention study in humans, but we have shown correlation that people with a higher blood pressure will have less of these good glycans. And very recently, only a couple of weeks ago, we published another paper showing that these bad glycans today predict hypertension in the future. So of course, we still have to do a lot of research. There is definitely not something like an FDA approved claim that if you improve your IgG glycans, you will have lower risk of cardiovascular events. But there is a lot of cross-sectional data suggesting that actually improving your glycans will improve your chances of not developing diseases. And of course, there is this entire line of evidence showing that inflammation is a predisposing factor for disease. And we know that these glycans are suppressing inflammation. So I think we can be pretty confident in claiming that if you improve your glycans, you are suppressing inflammation. And if you are suppressing inflammation, you're reducing the risk of cardiometabolic diseases. So I think it's pretty safe to claim that improving your GlycanAge is decreasing your risk of diseases, although of course we cannot make this type of claim because we don't have an FDA approval for that.
Yes, yes. Well, it's an exciting time in this whole space, and it'll be fascinating to see what comes down the line as far as diagnostic testing and as we understand these better. Knowing what you know as a world authority or the world authority on glycans and this space of longevity and health, I'd be curious if you would share with our audience how this knowledge has informed the personal choices that you make as far as your lifestyle and exercise and everything. Yeah, so knowing what you know, what do you do?
So that's a tough story. So I think 6 or 7 years ago, I learned that my GlycanAge is horrible. So currently I'm 51 chronologically, my GlycanAge is 78, which is really horrible. So first year, I decided, okay, I'm just too fat, I have to lose weight. So I spent entire summer hiking in the mountains, 3-4 hours a day, 20-25 kilometers hiking over the mountains. And I did not improve much. Because what happened, I did lose weight. I felt better, but I was overtraining. I had so much pain everywhere in my legs, in my feet. I was just doing too much exercise. And then I kind of gave up of overtraining. And then I was testing different things, including that I also discovered that I have this obstructive sleep apnea. So I have this CPAP now, which is making me sleep much better, which also improved my GlycanAge a bit. And something which was also scary for me, maybe 3 years after I had very high GlycanAge, I also developed hypertension. So I just went for a regular checkup and my blood pressure was all the way through the roof. It was something like 200 over 110. So I had an early warning, which did not help much, besides that I went for this checkup and discovered hypertension, which I now control with the pills. And definitely I use this GlycanAge to motivate myself not to eat too much, because I know when I gain extra 10 kilos, my GlycanAge goes up. I start losing kilos, my GlycanAge goes down. But I think my primary problem is my lifestyle. I work too much, it's too hectic, it's crazy. I have a lab of 50 people. I work at university, have 5 kids and a dog. So it's a lifestyle which it's kind of killing me. But I know, and I'm trying to improve. I'm always comforting myself that it would be worse if I would not be trying to improve. So we'll see. Maybe I wait for a little bit more research to be done with this colleague from New York, and maybe when I see which of everything he's giving to his patients is actually working, maybe I will also try that out.
It sounds good. Keep us posted, definitely. And Gordan, how can people get in touch with you, or or how can they follow you on social media and see what you're doing? Is there any website we could point them to?
So there is GlycanAge.com, which is a sales point for the GlycanAge. It's being sold globally, so anybody can buy it. I'm pretty active on Twitter and LinkedIn. I'm the only person with my name, so just Googling me on these social networks is fine. I also post a lot of on Facebook, but this is I do it on Croatian language in my home country, so this is not interesting for people outside. So I think Twitter and LinkedIn are the best places to see where they always post everything new what we do as a research on these 2 networks.
Great. Well, thank you so much for spending an hour with us today and helping us understand glycans and the fascinating world they are. It certainly opened up my mind, and I can't wait to follow the work you're doing and see all the new developments in the future. But thank you so much for being on the program.
So thank you for the invitation. One final note is that we are extremely interested in doing research and learning something new. So if anybody is doing an interesting study, any kind of intervention, trying to help people, we're interested to look into glycans.
Great, and please, and our audience, reach out to Gordan if you have any interest in that area. If you're on the scientific investigation side, that would be great. So thanks again, Gordan, and look forward. I hope this is the first of many conversations we have.
Thank you. It will be my pleasure.
Note: this is not intended to be a substitute for professional medical advice, diagnosis, or treatment. Always seek the advice of your physician or other qualified health provider with any questions you may have. Never disregard professional medical advice or delay in seeking of it because of something you have seen here. If you find this to be of value of you, please hit that like button and subscribe to support the work we do on this channel. Also, we take your suggestions and advice very seriously. Please let us know what you'd like to see on this channel. Thanks for watching, and we'll hope to see you next time!